Let's dig into the details of those two steps of how we went from prokaryote to eukaryote. So if we recall, the first thing that we need to do is to get an endomembrane system. And we're going to accomplish that with having the plasma membrane fold in on itself. In this case, we've got what's representing a prokaryotic cell. Here's the DNA as this little red string in the center. Remember that in a prokaryotic cell, one circular chromosome in the nucleoid region, not a true nucleus. We're going to get a true nucleus by having this plasma membrane fold in on itself. And if we have these parts come over here like this and pinch off, what we see then is that we have a nuclear envelope, right? DNA in the center, housed by 2 layers of phospholipid bilayer. So once we have this nuclear envelope that has 2 phospholipid bilayers, we can take that outer membrane and start to pinch it off, modify it, right? Make more phospholipid bilayers. We know how to do that, okay? And we start to get this folding. And now all of a sudden, we have things like an endoplasmic reticulum and a Golgi apparatus and vesicles and vacuoles and lysosomes and peroxisomes and all the other components of the endomembrane system. The second step in the evolution of prokaryotes to eukaryotes was the process of endosymbiosis. This is our proto-eukaryotic cell. It has the nucleus that has the 2 phospholipid bilayers making up the nuclear envelope, right? And we know that this outer membrane is going to become all of the components of the endomembrane system, which I didn't show for this diagram to make it simpler. Okay, but the next thing is that we need to get mitochondria and chloroplasts. So this process of endosymbiosis actually happened twice. Way back in the evolutionary past, there were prokaryotic cells that were really efficient at aerobic respiration. And at some point, one of these prokaryotic cells was engulfed into our proto-eukaryotic cell. Okay. This is actually how some organisms still to this day actually feed. It's called phagocytosis. Okay. They're going to bring in materials from the outside, pinch off a vesicle, right? And then this plasma membrane would actually pinch off, and these cut ends, right, are going to fuse with each other. I'm going to demonstrate that using my good old buddy Scotch tape. Regular phagocytosis, this vesicle would go and get fused with a peroxisome or something else that has the digestive enzymes to break down this prokaryote cell. But during endosymbiosis, this organelle was not broken down. Rather, its services were utilized in order to have the host cell, right, our main guy, production of ATP through aerobic respiration. We're going to go over the details of all that later in the course. So instead of breaking this down, we're going to kidnap it, and now it becomes ours. So, but this didn't happen just once. In fact, it happened twice. This particular prokaryotic cell was really good at doing the process of photosynthesis. Okay, so it comes along and gets engulfed by that host cell. This host cell, again, is going to pinch off a portion of its plasma membrane. So we can pinch off a portion of that plasma membrane to engulf this what used to be a prokaryotic cell, and now through the millions of years that after this has occurred, this is now a chloroplast, this is now a mitochondrion, and these organelles can no longer live on their own, but they maintain some of the characteristics that we see in prokaryotes. Step 2, boom, now we have true eukaryotic cells with a nucleus. It's membrane bound. We got 2 layers of phospholipid bilayers. We have membrane bound organelles making up the endomembrane system, and we have mitochondria with 2 layers of phospholipid bilayers, one from the prokaryotic ancestor, one from the eukaryotic host. Okay, same with our chloroplasts. We got some eukaryotic cells now, baby.